Biology lesson life process. We are going to learn 8 mark questions, 4 mark questions, 2 mark questions, and 1 mark questions. So, this video will be one short video. So, let's start. First question: how do you test the presence of starch in leaf? To write any activity, lab activity, these are the required side headings. So, you have to learn this: aim, apparatus, procedure, observation, result. So, let's start. You have to draw diagram also. Okay, let's start. Aim. I'm not going to highlight with any color because here already you can see in bold letters. So these are the highlighted words. So let's start. Aim. To show that starch is formed only in the green parts of a variegated leaf. Operators. Variegated leaf, beaker, water, spirit or alcohol, test tube, iodine solution, burner, burner forceps, drop. Procedure. So I'll explain procedure directly. You have to take variegated leaf from a plant. That plant has to kept in sunlight for few hours, and then you have to boil the leaf in water 1 to 2 minutes. You have to boil that leaf until it becomes soft, and then you have to keep that leaf in test tube. That test tube has to contain alcohol. Fourth point: place the test tube in hot water until the alcohol removes the chlorophyll and the leaf becomes pale. The leaf has to become pale in color. Fifth point, you have to wash that leaf with a warm water. With warm water, you have to wash that leaf. Sixth point, you have to spread that leaf in on a tile. Then you have to add iodine solution. You have to pour that iodine solution entire leaf. You can see here in this picture, A is before starch test and B leaf is after starch test. This green part is turned into blue color. Now we are going to see in this observation. Green parts of the leaf turn blue black. You can see in this picture. Point 2. Non-green parts do not turn blue black. They remain brown or yellow. Result: starch is formed only in the green parts of the leaf because chlorophyll is necessary for photosynthesis. Second point, non green areas do not form starch as they cannot photosynthesize. I hope you have understood first lab activity. Now, second lab activity. How do you prove that carbon dioxide is necessary for photosynthesis? Aim: to show that carbon dioxide is needed for photosynthesis. Operators: a carbon, a healthy potted plant, bell jars, 2 bell jars, KOH, potassium hydroxide, Vaseline, iodine solution, dropper, glass plate, matches. So you can see here CO2 means carbon dioxide, KOH means potassium hydroxide. Wherever you see this, you have to remember. Okay, procedure. Keep the potted plant in darkness for 24 hours to discharge discharge the leaves. Second point, you have to select 2 leaves from the plant that has to be attached to the plant itself. Third point, cover each leaf with a separate bell jars. You can see here one bell jar and this is another bell jar. Place a disc containing KOH solution inside one bell jar. This is KOH solution. In one bell jar it is present and in another bell jar it is not present. KOH solution is not present. So see here KOH absorbs carbon dioxide completely. Fifth point, do not keep KOH in the second bell jar. Sixth point, seal the bottom of the bell jars with Vaseline to make them airtight. Keep the setup in sunlight for a few hours. Eighth point, after this, Remove both leaves and test each leaf for starch using iodine solution. You have to test each leaf. Observation: the leaf inside the bell jar with KOH means no carbon dioxide does not turn blue black. The leaf in the bell jar without KOH means in that carbon dioxide is present, so it turns blue black. Result: only the leaf that had carbon dioxide shows starch formation. Therefore, carbon dioxide is necessary for photosynthesis. Third question: write flow chart showing breakdown of glucose molecule by various pathways. So, breakdown of glucose by various pathways. The breakdown of glucose occurs in three main pathways. These are the three main pathways depending on the presence or absence of oxygen. Glucose takes place in cytoplasm. The 6 carbon molecule is breakdown into 3 carbon molecules. So that is only pyruvate. This pyruvate releases some energy. These are the subsequent pathways. These three are the subsequent pathways. First pathway: absence of oxygen in organisms like yeast, and this is converted into means pyruvate is converted into ethanol, carbon dioxide, and energy. Lack of oxygen in human muscle cells. Okay. Then pyruvate is converted into lactic acid plus energy. Presence of oxygen in mitochondria. Then pyruvate is converted into carbon dioxide plus water plus energy. So this is the flow chart. You have to write. Fourth lab activity. This is same as first lab activity, so I'm not going to explain. So read this once. Fifth lab activity. Conduct an experiment that light is necessary for photosynthesis. Aim to show that only the parts of a leaf that get light can make starch. Apparatus: potted plant, black paper or foil, tape, water, alcohol, burner, iodine solution. Procedure: keep the plant in darkness or for 1 day to remove starch. Second point: cover one part of leaf with black paper so that light cannot reach it. So this is black paper strip. Keep the plant in sunlight for a few hours. Pluck the leaf and boil it in water. You have to pluck it means you have to pluck it. Pluck the leaf in boiling it in the water. Then boil it in the alcohol to remove green color. Wash it and add iodine solution at last. Now observation. The part of the leaf exposed to light turns blue black. The covered part does not turn blue black. So whatever the covered part means black paper strip is there na, that part does not turn blue black and remaining part will turn into blue black means that part is exposed to sunlight, so it turns into blue black, and remaining part means which is not covered part, that does not convert into blue black. Result: only the part that got light made starch. Therefore, the light is necessary for photosynthesis. Now, sixth question: how do you distinguish or differentiate between Arteries from veins. Arteries and veins. Arteries that carries blood away from the heart, carry blood towards the heart. So this is away from the heart, this is towards the heart. Usually carry oxygen rich blood. Usually carry oxygen poor blood. Have thick elastic walls, have thin less elastic walls. Blood flows under high pressure. Blood flows under low pressure. No walls present. Walls are present to prevent backflow. To prevent backflow, walls are present. Have a narrow lumen. Have a wide lumen. Okay, lumen. L- Lie deep inside the body, often lie near the surface. Blood sprouts when cut. Blood flows slowly when cut. You can see arteries carry blood away from the heart. Veins carry blood towards the heart. Away from the heart, towards the heart, carry oxygen rich blood. This is oxygen poor blood. Thick elastic walls. This thin less elastic walls. Blood flows under high pressure. This is low pressure. No walls present. Here walls are present because to prevent from backflow. Narrow lumen. This is wide lumen. In this is lie deep inside the body. These often lie near the surface. Blood sprouts when cut. This blood flows slowly when cut. Seventh question. Explain the stages of nutrition in amoeba by the following diagram. You can see this diagram from this you have to write. Stages of nutrition in amoeba. So first see the stages. Okay, first see this. Amoeba shows holozoic nutrition. The nutrition process takes place in five main stages. So you can see five main stages. Ingestion means taking of food, digestion, breaking down food, absorption, taking in nutrients, assimilation, using the absorbed food, digestion, removing undigested food. These are the five stages. First stage, ingestion, taking in food. 3 points is there. Amoeba senses the food particle, forms pseudopodia, means finger-like extensions around the food. The pseudopodia enclose the food and bring it inside the cell. These are the 3 points. Second stage, digestion, breaking down food. After ingestion, a food vacuole, vacuole is formed around the food particle. Digestive enzymes are released into the vacuole. These enzymes break the complex food into simple soluble substances. Third stage, absorption, taking in nutrients. The digested food is absorbed from the food vacuole into the cytoplasm. The absorbed food is used for growth, energy, and other life activities. Fourth stage, assimilation, using the absorbed food. The absorbed nutrients are used for creating new cell material, repairing parts, and producing energy. Fifth stage, digestion, removing undigested food. The undigested waste remains in the food vacuole. The food vacuole moves to the cell membrane, and last is waste is expelled out by the rupturing the memory. sorry, membrane. So these are the five stages. So what are these stages? Ingestion, digestion, absorption, assimilation, digestion. These are the five stages in amoeba. So I'm going to say this in short, in only one line or two lines. That's it. Okay, please wait. So see here, ingestion means taking in food, means the amoeba detects a food particle in its environment and then it captures. It extends temporary finger-like projections like pseudopodia to surround the food and then enclosure. These pseudopodia fuse, trapping the food and a small amount of water inside a food vacuole. Second stage, digestion, means breaking down food. The digested soluble nutrients, sorry, digestive enzymes from the surrounding cytoplasm are released into the food vacuole. Then it breaks down the enzymes chemically, means complex food particles into simple soluble molecules. Now, third stage, absorption, taking in nutrients. The digested soluble nutrients passes directly out of food vacuole and they turn into the cytoplasm. These nutrients are then spread throughout the entire cell. Fourth one, assimilation, using the absorbed food. The amoeba uses the absorbed nutrients from for several vital functions like growth and cell repair, creating new cell material, and then last function is respiration to produce energy for life activities. Last stage, it is digestion, removing undigested food. Any food material that cannot be digested remains inside the food vacuole. The food vacuole moves to the edge of the cell, fuses with the cell membrane, and ruptures through the undigested waste out of the body. Means undigested waste remains in the food vacuole. The food vacuole moves to the cell membrane. Waste is expelled out by rupturing the membrane. Now eighth question. Write the differences between autotrophic nutrition and heterotrophic nutrition. Autotrophic nutrition, heterotrophic nutrition. Organisms make their own food. Organisms depend on others for food. Own food and here depends on other for food. Uses sunlight, carbon dioxide and water to prepare food. Uses ready-made food from plants or animals. Found in green plants and some bacteria. Found in animals, fungi and many bacteria. Food is made by photosynthesis. Food is obtained by ingestion and digestion. So organisms make their own food. Here depends on others For food, uses sunlight, carbon dioxide, and water to prepare food. Here, uses ready-made food from plants and animals, found in green plants and some bacteria. Here, animals, fungi, and many bacteria it is found. So, food is made by photosynthesis and here obtained by ingestion and digestion. Ninth question. What are the steps involved in the process of photosynthesis? Steps involved in the photosynthesis. These are the steps. Four steps you can see. First step. First step, absorption of light. Second step, absorption of carbon dioxide and water. Formation of glucose. Release of oxygen. Steps involved in photosynthesis. Photosynthesis occurs in green plants and takes place mainly in the chloroplast. It involves the following main steps. You have seen these side headings, right? First stage, first step, absorption of light. Chlorophyll in the chloroplast absorbs sunlight. Here, absorption of sunlight happens, and then sunlight provides energy needed for photosynthesis. Second stage, absorption of carbon dioxide and water. Leaves take carbon dioxide (CO2) from the air through stomata. Through the stomata, carbon dioxide is taken. Roots absorb water (H2O) from the soil. Third step, formation of glucose. Using the energy from the sunlight, carbon dioxide and water are converted into glucose C6H12O6 in the chloroplast. Fourth point, release of oxygen. Oxygen O2 is released as a byproduct into the air through the stomata. Chemical equation of photosynthesis you can see here. 6 CO2 plus 12 H2O gives rise to, and conditions are sunlight and chlorophyll. These are the um, then glucose C6H12O6 plus 6O2 and plus 6H2O. This is the chemical equation of photosynthesis. This is very important. So learn this properly. So learn properly. 10th question. Draw a neat label diagram showing cross section of leaf. So practice this diagram. Practice labeling also when you are practicing diagram. So labeling is also important. 11th question. Observe the given diagram and answer the following questions. What does this picture indicate? Answer stomata. Identify the parts A and B. Answers A guard cells and B is chloroplast. Here should be A and here should be B. So answers are guard cells and chloroplast. What happens if this organ is not present in the leaf? Answer, if stomata is absent, then carbon dioxide cannot enter, oxygen cannot exist, and photosynthesis and also transpirations are greatly reduced. Okay, 12th question. Observe the following given diagrams means diagram and answer the question. What does the given diagram represent? Nutrition in amoeba. Answer is nutrition in amoeba. Identify the parts A and B. A and B. A is nucleus and B is food. What are the finger-like projections in the picture called? Pseudopodia. Before also we have learned this. 13th question. What questions would you like to ask a dentist? Answer, you have to write these. Whatever questions you're going to ask, you can write. For example, how often should I brush and closes my teeth? What is the best way to prevent cavities? How can I keep my uh, gums healthy? Are sugary foods really harmful to teeth? What should I do if I have a tooth pain or sensitivity? How often should I visit a dentist for a checkup? How often should I visit a dentist for a checkup? What are the causes of bad breath and how can I prevent it? How can I take care of my teeth at home besides brushing? So these are the questions you have to ask to a dentist. 14th question. What questions would you like to pose if you get a chance to meet a cardiologist? These are the questions you have to, if you visit a cardiologist, whatever you are having in mind, you can write here. See, how can I keep my heart healthy? What are the early signs of heart disease? How much exercise is good for the heart? Which foods are best for heart health? How can I reduce high blood pressure? How often should I check my heart health? What are the common causes of heart attacks? Can stress affect heart health? These are the questions. 15th question. Write the slogans to create awareness on organ donation. So these are the slogans you can learn. I think. 16th question. Now you can take screenshot and learn the slogans. 16th question. What are the differences between combustion and respiration? This is the difference between combustion and respiration. First, first point happens fast. This slowly. Needs high heat. Normal body temperature is enough. Happens outside cells. Inside cells. Burns fuels like wood or petrol. Breaks down glucose. Pros- um, Produces carbon dioxide, water, and lots of heat. Produces carbon dioxide, water, and energy. Here, lots of heat, here it is energy, ATP. Energy comes all at once. Here, gradually, energy comes. Can see flame, heat, smoke, not visible directly. Does not keep living things alive. Provides energy to living things. These are the differences between combustion and respiration. 17th question. What are the differences between uh, aerobic and anaerobic respiration? Aerobic and anaerobic respiration differences we are going to learn now. Aerobic respiration requires oxygen. Here does not require oxygen. Takes place in mitochondria in cytoplasm. Glucose completely breaks down into carbon dioxide and water. Glucose partially breaks down into lactic acid or ethanol plus carbon dioxide. Releases large amount of energy 36 to 38 ATP per glucose. Here anaerobic respiration releases small amount of energy that is 2 ATP per glucose. End products are carbon dioxide and water. Here end products are lactic acid or ethanol plus carbon dioxide. Occurs in plants and animals under normal conditions. Here occurs in muscles during strenuous exercise or in yeast or fermentation. Seventh point. Energy release is slow but efficient. Energy release is fast but less efficient. Eighth question. No harmful by byproducts accumulate. Here byproducts like lactic acid can cause fatigue. 18th question. So by seeing this flowchart you have to answer the following questions. Question A. What does the flowchart indicate? Answer: breakdown of glucose by various pathways. Fifth, B. Name the place where lactic acid is formed. You can see here lactic acid in human muscle cells. C. What is the starting factor in this Reaction. Starting factor is glucose. Name the products formed after the reaction in mitochondria. Here mitochondria is there. So what are the reactions? So carbon dioxide, water and energy. Carbon dioxide, water plus energy. Completed 18th question. Now 19th question. What type of questions you would like to ask if you get a chance to meet a gastroenterologist? Okay, gastroenterologist. First question. How can I keep my digestive system healthy? What foods should I eat or avoid for good digestion? What are the common causes of stomach pain or acidity? How can I prevent constipation or diarrhea? How often should I get my digestive health checked? What are the early signs of liver or stomach disease? How does stress affect digestion? What should I do if if I have frequent indigestion or heartburn? These are the questions. So then, question 20. How do plants manage their waste? How do plants manage their waste? Some waste are stored in vacuoles inside cells. Waste like water, oxygen leave through leaves. Waste can be stored in old leaves, bark, or wood. Plants release some waste as gums, resins, or latex. Some waste are converted into useful substances for the plant. So now 2 mark questions. 21. Write equation for photosynthesis. Before we have learned, so learn this formula. Means equation. Second, 22. What is the role of hydrochloric acid in human digestive system? Kills harmful bacteria present in food, helps digestion of proteins by activating the enzyme pepsin. Okay. Next, what does 120 by 80 indicate? The numbers 120 by 80 represent normal blood pressure. 120 means systolic pressure, means pressure in the arteries when the heart beats. 80 means diastolic pressure, pressure in the arteries when the heart rests between beats. So 120 by 80 means normal blood pressure. 24th question. Read the following passage and write answers for the given questions. Okay, paragraph is missing, but you can see directly answer. What changes happen when tooth decay starts? Cavity forms. When are acids released? Acids are released when bacteria digest sugar from food. Then bacteria is released. How is plague formed? Plague forms from bacteria, food particles, and saliva on teeth. Fourth, what should we do when we get dental caries? Dental caries. Visit a dentist for treatment. 1 mark questions. What are the materials required for photosynthesis? Carbon dioxide, water, sunlight, and chlorophyll. Next, the xylem is responsible for dash plants. Answer is A. Transport of water. What is nutrition? The process by which living organisms take in food and use it for energy, growth, and repair is called nutrition. Take in food and use it for energy, growth, and repair is called nutrition. Next, digestion does not occur in which part of alimentary canal? Answer, esophagus. Esophagus does not occur in alimentary canal. Okay. Expand ATP, adenosine triphosphate, adenosine triphosphate. What is the instrument used by doctors for measuring BP? It is sphygmomanometer. Sphygmomanometer. What is plasma? The liquid part of blood that carries cells and nutrients is called plasma. Finger-like projections in small intestine are called villi. Why are cartilaginous rings present in the trachea? Trachea. Okay. To keep the trachea open for air passage. To keep the trachea open for air passage. Air passage is happening when you keep the trachea open. 34th. Which one will have longer small intestine? A goat or a tiger? Why? Answer is goat because herbivores need longer intestines to digest plant food. What will happen if HCl is not secreted in the stomach? Proteins cannot be digested properly. Next, what will happen if the respiratory tract is not moist or there is no mucus? Answer, dust and germs will enter causing infections. Next, what will happen if the platelets are not present in the blood? What happens if platelets are not present in the blood? Answer is blood cannot clot properly and bleeding continues. Blood cannot clot properly and bleeding continues. When platelets are not present in the blood, these causes. Answer is blood cannot clot properly and bleeding continues. What is the digestive juice without enzymes? It is bile juice. So 38 questions we have learned. Okay, these questions we have covered in this video. So now I'm going to provide you some few questions. 8 marks, 4 marks, 2 marks, and 1 mark question. So see here from this is also first lesson. 8 mark questions. First question. Explain the structure of nephron with diagram. See you have to draw this nephron image or sorry, picture. You have to draw this picture. Okay. Draw this diagram. You have to draw nephron diagram and you have to explain it. Explain about the nephron. So a nephron is structural functional unit of kidney. Unit of the kidney. Each kidney has about 1 to 1.5 million nephrons. It filters blood and forms urine. Okay. Main parts of a nephron. Bowman's capsule. Glomerulus. Okay. Glomerulus. Proximal convoluted tubule. PCT. Loop of Henle. Distal convoluted tubule. DCT. Collecting duct. So these are the main parts of nephron. You can see in this picture also. Bowman's capsule. Cup-shaped structure that receives blood filtrate from the glomerulus. Second part, glomerulus, a network of capillaries inside Bowman's capsule. Filters blood under pressure, means ultrafiltration. Third part is proximal convoluted tubule (PCT), a long coiled tube after Bowman's capsule. Reabsorbs water, glucose, amino acids, and salts. Fourth part, loop of Henle, hairpin shaped structure with a descending and ascending limb. Helps concentrate urine. So, in short, filtration, reabsorption, secretion. Fifth point, distal convoluted tubule (DCT). Further reabsorption of salts and water, controlled by hormones. Collecting duct collects urine from many nephrons. Collects urine from many nephrons. Final concentration of urine occurs here. Okay. Filtration, reabsorption, secretion. You can learn this question. Okay. Properly learn this question. Second question. How are lungs designed in humans for exchange of gases? First, see the side headings. Large surface area, thin alveolar walls, rich blood supply, moist surface, efficient ventilation, elastic lung tissue, branching air passages, means passages, branching air passages, maintained concentration gradient. These are the side headings and learn about this here. From here you can learn. So I'm moving to third question. Describe the double circulation in human beings. Why should we do it? Why should we need it? See, the double circulation in human beings. Okay, this is double circuit circulation in human beings. First, see the diagram. See the arrow marks also. Okay, now answer. Blood passes through the heart two times in one complete cycle, so it is called double circulation. Pulmonary circulation carries deoxygenated blood from the right ventricle to the lungs through the pulmonary artery. You see the highlighted part. I'm going to read only the highlighted part, and remaining you can read on your own. In the lungs, blood becomes oxygenated. This oxygenated blood returns to the left atrium. Systematic uh, circulation begins when the left ventricle pumps oxygenated blood. So blood cells use the oxygen and nutrients. This deoxygenated blood returns into the right atrium. Need of double circulation. Double circulation is needed to keep oxygenated and deoxygenated blood separate, which increases the efficiency of oxygen supply required for high energy needs in humans. Fourth question. What would be the consequences due to deficiency of hemoglobin in our body? Consequences of globin deficiency. What are the consequences of hemoglobin deficiency? We are going to learn now. Less oxygen reaches body cells, tiredness and weakness, shortness of breath, pale skin and pale lips, dizziness or fainting, faster heartbeat, poor concentration, overall reduced immunity, and poor health. These are the consequences. Fifth question. How is food transported in plants. So learn this. 4 more questions. Sixth question. How is small intestine designed to absorb digestive food? Long and coiled, inner walls have villi have blood vessels. Thin walls. Okay. Learn now. I have said only the side headings. Now you can learn from here. Okay, what is mean by long and coiled? The small intestine is very long, giving more time and space for food to be absorbed. So see this, learn this question answer. Seventh question. What are the differences between lymph and blood? First see blood and lymph. Red colored fluid. This is colorless or pale yellow fluid. Contains contains RBCs, WBCs, and platelets. It contains WBCs only. Okay. Here RBCs, WBCs, and platelets. Here only the WBCs. Flows in blood vessels. Flows in lymph vessels. Transports oxygen, nutrients, hormones, and waste. Here helps in immunity and returns tissue fluid to the blood. Blood pressure is high. Here is slow and pressure is low. Flow is slow and pressure is low. Here blood pressure is high. Here pressure is low. Eighth question. What is the difference between exlim and phylum? Exlim and phylum. You can learn from here. Mm, now ninth question. How is oxygen and carbon dioxide transported in human beings? Transportation of oxygen, transportation of carbon dioxide. Oxygen enters the blood in the lungs. It combines with hemoglobin in RBCs. Blood carries oxygen to all body cells. Now, transport of carbon dioxide. Carbon dioxide from cells enters the blood. Most of it is carried in the plasma. Blood brings carbon dioxide back to the lungs to be exhaled. So, this is the process. Okay. How is oxygen and carbon dioxide transported in human beings? This is the answer. So, 10th question. What are the similarities between um, aerobic and anaerobic respiration? So, before we have learned the difference, right? Now we have to write similarities. Both break down glucose to release energy. Both start with glycosim, gly, glycosis in the cytoplasm. Both produce ATP, means energy. Both produced by byproducts like carbon dioxide or other products. So breakdown of glucose, uh, glycosis in cytoplasm and produce energy, produces byproducts. Okay, these are the similarities between aerobic and anaerobic respiration. 11th question. What questions would you like to pose if you get a chance to meet a pulmonologist? Pulmonologist. Okay. What are the questions? These are the questions you can learn from here. This is also question based. What what questions would you like to ask if you get a chance to meet a nephrologist? So learn this. Okay. 13th question. Raju is a 13 years old boy and only 22 kgs only. The weight is not sufficient. Which doctor he has to meet and what type of questions he should ask. So these are the questions. Learn this. 14th question. What is the role of saliva in the digestion of food? This is very important. Saliva moistens the food, making it easy to chew and swallow. It contains an enzyme called amylase. Amylase breaks down starch into sugar. Amylase breaks down starch into sugar. Saliva also helps clean the mouth and protects the teeth. Protects teeth. So moistens the food. Amylase. It contains amylase. Break down starch into sugar. Clean the mouth and protect teeth. These, this is the role of saliva in the digestion of food. 15th question. How the plants get rid of their waste materials? Leaves, stems, roots, fruits and flowers, excretion in another forms. 16th question. Why is it necessary to separate oxygenated and deoxygenated blood in mammals and birds? Mammals and birds are warm blooded and need lots of energy. Warm blooded and lots of energy needed. Oxygen is required for cellular respiration to release energy. Separating oxygenated and deoxygenated blood ensures efficient oxygen supply to blood cells. It helps maintain high metabolism and supports active lifestyles. So, warm blood, lots of energy. Cellular respiration, efficient oxygen supply, high metabolism, and it supports active lifestyle. Now, we are going to move to marks questions. We are going to learn marks questions. These are the extra questions, but you have to learn these all. 17th question. Define nutrition. What are the different types of nutrition? Nutrition means we have learned previously also, so I'm not going to discuss here. Just briefly, I'm going to discuss. There are two types of nutrients: autotrophic nutrition, heterotrophic nutrition. Autotrophic means organisms make their own food. Heterotrophic nutrition means organisms depend on others for food. For autotrophic nutrition, example plants. Heterotrophic nutrition, uh, heterotrophic trophic nutrition example is animals. 18th question. Why do carnivores have shorter small intestine? Carnivores eat meat which is easy to digest. So they have short small intestine to absorb nutrients quickly. 19th question. What is fermentation? Give two examples. Fermentation is a process in which microorganisms break down food to release energy without using oxygen. Without using oxygen, if the food is break down and released energy, then it is called fermentation. Example: yeast ferments sugar to produce alcohol and carbon dioxide. Bacteria ferment milk to make curd. Question 20. Name the processes you consider essential for maintaining life in living beings. So these are the essential life processes: nutrition, respiration, transportation, excretion, growth, reproduction, response or movement. Question 21. What would be the consequences due to deficiency of platelets in our body? Sorry, in our blood. Platelets in our blood. First, blood cannot clot properly, leading to excessive bleeding and slow healing. Easy bruising and risk of internal bleeding may occur. 1 mark questions. Give an example for parasitic nutrition in plants. Parasitic nutrition in plants. Kaskuta. ADP plus dash ATP. ADP plus PI is equal to ATP. One ATP molecule release dash energy in kg. One ATP is similar to 30.5 kg. How many carbon atoms are in each pyruvate molecule? So we have learned in the flowchart. 3 carbon atoms. Name the different types of heterotrophic nutrition. Parasitic, saprophytic, Mesozoic, symbiotic. These are the 4 examples. 4 types of heterotrophic Nutrition. What are blood sucking animals called? Give example. Leech and mosquito. What type of chemical reactions takes place in our body to break down the food materials? Answer. Catabolic reactions or hydrolysis. 29. Herbivores. Herbivores. Very long intestine. Carnivores. See here. Answer missing is short, small intestine. 30. Kidneys are essential organs for survival. How do you support your statement? Answer. Remove waste, maintain water and salt balance. So learn remaining questions from here. Okay, complete. So we have covered 8 marks, 4 marks, 2 marks and 1 marks from lesson 1. That is only life process. From life process we have covered all. So learn this question and answer. So I hope you will like this video. So thank you. Thank you for watching.